wrap up on antibodies function, and we start looking at those fun gene rearrangements for lymphocyte development. That is like cool stuff. Okay. All right, so we stopped in this slide last time, right? So basically looking at different classes of antibodies. And here is showing you like the percentage or how much of them do you have in your circulation, right? So this is your blood serum. And we see that the IgG, which is that monomer, right? The IgG, you have several types, also several classes of IgGs, but those are the most abundant in your bloodstream, okay? Then the following one is IgA, and also if you look at IgA that is in the blood circulation, you're going to find the monomer, right? Because you are going to have the dimer IgA when you get to the mucosal tissue, but in the circulation, it circulates as monomer, okay? And then the third most abundant is IgM, and that guy is your pentamer when it's secreted, right? So IgM is monomer only when it is at the surface of the B cell. So when the B cell shoots out the antibody as a secreted protein, then this thing is a pentamer, right? And one very interesting and important thing about IgM, this is the first class of antibodies that show up once you are exposed to a certain pathogen, right? And also especially when your innate system did not totally eliminate the pathogen. So after exposure to a pathogen, you are going to start activating your adaptive system, and then you're going to start producing antibodies, and IgM is the first class that gets produced, okay, first class that gets secreted. So keep in the back of your mind that when you are looking for recent active infections, you are going to be looking for what type of antibodies you have in the blood. So if you have increased production of IgM against a specific antigen, right, or against a specific pathogen, then that is a telltale sign that you have a recent and active type of infection going on. Okay, so high level of IgM means recent infection. Okay. Then you have this other guy here that is IgD. And IgD, basically, it doesn't get secreted. It doesn't really have a function outside of the membrane of the B cell. Okay, so the main function for IgD is to direct the mature B cell to come out of the bone marrow and to go to secondary lymphoid tissue. Okay, so that's the main function of the IgD. So IgD is expressed on the surface of your B cell that is mature. So here is your B cell, okay, mature B cell. And after it goes through the process of selection and rearrangement, right, in the bone marrow, this mature B cell is going to have on the surface is going to have two types of antibodies. It's going to have IgM and is also going to have IgD. Okay? So those two are mature B cell receptors. But the cell itself, this B cell here, although it is mature, it is still naive. It did not see antigen. So it needs to migrate to the secondary lymphoid tissue, and over there it's going to see antigen. But in order for it to get there, IgD is important for conducting the travel arrangement for this B cell. Okay, so that's the function for the IgD. And now IgE is a class of antibodies that is at lowest concentration in the plasma. And usually that's the case unless you have very serious problems of allergies, because IgE is very important against helminth types of infections, so against those worms, right? But since we are not really fighting a lot of parasitic worm infections in more developed countries, IgE starts to pick up some other annoying type of functions, like make your immune system a bit overreactive and give you some allergies. So IgE is the guy, responsible for allergic reactions, the bad types of allergic reactions. Okay, and this goes hand in hand with that hygiene hypothesis. Have you heard of that one? No? Hygiene type of hypothesis? Oh, basically, when you study the immune systems of people that are growing up in third world countries or not very developed countries, you see that first they are exposed to a lot more pathogens than we are at a place that life is just different. We're not really concerned about pathogens that you find like in the dirt or in the soil, because we are usually walking around with shoes everywhere, right? We don't step on things and then things crawl under your skin. You know what I'm talking about? Yeah, right? Or maybe not, right? Now, actually, I do have one experience growing up in a society like here, and interestingly enough, there was also a case I could also observe when my older son was born. Okay, so I have two sons. They both come here. Adam is 25, and Alex is 20, is almost 21. So anyway, when the first one was born, so I had this paranoia about keeping everything very clean, like everything had to be sterilized, pacifiers, I boiled them first, the kind of thing. Maybe you don't know the kind of thing. Anybody has kids in here? No, so we will find out about it. Okay. Or if you have friends who are going through this stage already, oh yeah, you get totally paranoid. It's like the baby is crying, oh my God, something's happening, Oh, the baby is hungry, I don't know why the baby is crying. No, that believe me, it happens. If you are a caring parent, some parents are like, oh, what the hell, he'll shut up. But no, I was like totally paranoid. And then what happens? My older son started to develop asthma. It was not fun. I was lucky that at the time I was living only like 4 blocks from Joe DiMaggio, because I tell you, I could run that path in less than 1.5 minutes to get to the ER when that baby was not breathing and turning blue. And that was asthma, okay? Bad asthma. So by the time he was 4.5 or so, then we had our second son, And with the second son, I think I got too tired of cleaning up the house all the time. He's always getting dirty anyway. So yeah, I mean, it's not dirty, but I wasn't cleaning every other day, scrubbing the floors, And Alex was exposed to a lot more environmental diversity, I like to put it that way, right? Than Adam was. Right? And foods also exposed to a lot of foods before, the age that the doctor says this is appropriate or not appropriate. Like, for example, we had those double strollers, right? So we go in the zoo and my older son wants an ice cream and he gets an ice cream. And the 6-month-old baby is looking at the ice cream and going, that looks yummy, So then Adam goes, oh, I think you want ice cream. You want ice cream? And there goes the introduction to the ice cream. Instant. Yeah, my poor second child, he got a lot more exposure. Never had allergies. Never had asthma. He had a much stronger immune system. And there are other factors that go with that too, but that's a different story. But yeah, it's kind of amazing, right? So get ready when you have kids. All right, so coming back here, IgE. If all this session, this third type of immune response of the immune system, type 3 or so, if it doesn't get a lot of stimulus, then the IgE starts to get produced against things that are not harmful for you, like allergies, pollen, and stuff like that. Okay. All right. So here, the idea of antigens and epitope. So these are just terms that you have to know. Okay. Antigen. What is an antigen? Is something that is going to be recognized by antibodies. That is going to cause a reaction. It's going to cause an immune reaction. So here you have a whole protein or a whole thing that this thing is an antigen. that when it gets to your system, you're going to have some type of immune reaction, and you're eventually going to produce antibodies. Okay, that's the whole thing, is the antigen. Now, when you talk about epitope, those are little fragments of your antigen that are specifically recognized by one antibody. Okay, so you have a little piece here, let's say that is about 8 amino acids long, usually 8 to 10 to 12 amino acids long. So this little piece here is recognized by one antibody. So that is an epitope. So when you look at an antigen, the antigen can have several epitopes. So the bigger the antigen, the more epitopes it can have. Okay? Now, besides this antigen and epitope idea, you also have this idea here, the notion of something that is called a hapten. Okay? So a hapten is a very, very small epitope, but is not big enough to produce allergic or immune response. So a hapten is going to be like something tiny, but by itself, if you introduce a hapten, like by injection in your body, by itself, you are not going to develop antibodies against that. Okay, so in order for a hapten to develop, for you to develop antibodies against the hapten, it will have to be connected to another piece of, usually of a protein, that is called a carrier protein or a conjugate protein. Okay, so now you're going to have a bigger thing, and this bigger thing is going to allow you to develop antibodies against that hapten. Okay, so hapten is a tiny epitope that doesn't really allow development of antibodies. Okay. All right, so you can potentially develop antibodies against each of the epitopes on the antigen, right? So let's say that I have a big protein or an inactivated virus or a non-replicating virus, and let's say that you get injected like a vaccine with the virus. Okay, so if you get injected with the whole thing, so basically your body is going to produce antibodies against individual antigens, epitopes on your antigen. Okay, I can give you epitopes on your antigen. So when you look at all the antibodies against the antigen, you might have all different types of antibodies that recognize different pieces of your antigen. Okay? When that happens, you say that this whole soup of antibody that you have, so now let's say that you have this sequence of this antibody over here. Okay, so let's say that you have cloned the sequence of this antibody in a vector, and you are going to go to the lab and you're going to produce that molecule. So you're going to produce this protein in the lab, and you are going to end up with a vial here that has a lot of antibodies that are only of this kind that you have produced. That is one specific clone of an antibody. So you call that a monoclonal antibody. Okay, so only one type of antibody that is getting produced. Okay, and now you're thinking, how is it that the antibody is interacting with the antigen? Okay, you're going to have areas at your binding sites. Those are the variable regions. Remember, these binding sites are exactly the same as the other arm of that antibody. Okay, and these are going to interact with, let's say this thing is a protein. So it's going to interact with the protein. Okay, the interaction that is happening, it's not a very strong interaction. You're not going to form covalent bonds. Okay, so type of interaction that you're thinking about is basically hydrogen bonding. Okay, we're also talking about hydrophobic interactions. Okay, so those fall under these categories here. And you also have some types of electrostatic interactions. How do you get electrostatic interactions? Everybody took cell bio? Yes? Anybody took cell bio with me? Yeah, one person. Okay. How?
Different charges?
Yeah, Yes, different charges. So you have here protein, right? You have here a protein. Is antibody a protein? Yes. So here is your hand of the antibody before the armpit, that is the hinge region right there. So this is only one arm of the antibody. So here you're going to have different charges, okay? And then when you look at your antigen, you're going to have different charges too. They're going to interact. So those are your electrostatic types of interactions. This is going to be like a perfect fit of your binding site of the antibody with the recognized part of your epitope. Okay, so these interactions here, they are transient. They can bind and release, right? So they're not crazy glue type. Okay, bind and release, and but they're usually very, very specific. Actually, that's the game for antibodies. They have to be very specific. Okay, to the antigen. All right. So that brings us to this other idea here of how is it that the antibodies bind to what they recognize. Okay, so you have these two other definitions that you absolutely have to know. One of them is affinity, the other one is avidity. Okay, easy to remember which one is which. Okay, if you have an antibody that binds like strongly with the antigen, then you can say that it is grabbing it really strongly. It has a good affinity for that antigen. Okay. So the higher the affinity, the stronger it's going to bind. So how strongly or how the antibody grabs the antigen, that is your affinity. Now, when you look at the possible binding sites of antibodies, that is going to be your avidity. Now, what does that mean? If you look at a monomer, monomer has two binding sites, so it has an avidity of 2. So if you look here at a dimer, so a dimer has 2 antibody molecules. How many binding sites does it have? 4. So what is the avidity of your dimer? It is 4. That is not telling you how strongly it's binding to the thing, but it tells you it has avidity of 4. And what about your pentamer? It has 10. Okay, 10, avidity of 10. 10 possible binding sites for that antigen. Every one of these binding sites is exactly the same. Okay, so this guy here that is IgM, every binding site binds exactly the same epitope. Okay, that's an important idea to have. Okay, because this thing is produced by one cell that is one clone, as we're going to see how the clones develop. Okay. All right, so everybody cool with affinity and avidity? Yeah?
Can you increase affinity by increasing avidity? Or are they separate things?
They are separate things. But you do increase affinity. The avidity is just intrinsic property of how many arms the antibody has. But the affinity changes depending on the infection type and depending on stimulation of T helper cells. Okay, so especially when you're looking at IgG antibodies, those are going to have higher affinities. Okay, but the avidity is always the same. IgG is monomer is going to have avidity too. Okay, so the first antibody that you produce, as we mentioned, is your IgM, right? So IgM is going to have your highest avidity, but actually IgM has lowest type of affinity. So let's say that after IgM, you're going to have class switching from IgM to IgG. So the IgG is going to be with higher affinity. Okay, I was going to see that, how that happens. All right, so here more on how antibodies are working. So we're briefly looking at the functions of neutralizing the bacteria or the viruses or the toxins, right? And besides neutralizing, it can block the binding of those pathogens, right? So here those are those functions. And here another function, of course, that is very important is the opsonization of pathogens, right? So antibodies can recognize epitopes on pathogens, and the constant region of the antibody has receptors on phagocytic cells that can go there and eat up the pathogen, the bacterium or the virus. Okay, so this is the opsonization area of pathogens, of antibodies. All right. And also I mentioned that antibodies work with red blood cells, right? So remember that you have several ways that red blood cells can work in immune system clearance of pathogens, right? Remember that red blood cells, they have those CR1 receptors that pick up antigen, that pick up a complex of antigen and antibodies. Okay, so CR1 can interact with antigen-antibody complex, okay, and clear up those complexes. Okay, remember that if you have immune system activation by antibodies, you're going to have the classical pathway activation, and the antibodies that are important for classical pathway, remember those are IgM and IgG. Okay. So besides this function of antibody-antigen complex, you have another function here for your red blood cells. So red blood cells can have these receptors that bind to the constant region of antibodies. So those receptors are called Fc receptors. Okay? So Fc receptors, mainly they are going to bind to antibody class IgA. Remember, IgA in blood is mono. So Fc receptors also can help for clearance of IgA-bound antigen. Okay, and there are some interesting things also about antibodies binding to red blood cells. So there are some diseases. Okay, this is a new updated slide. So there are some diseases that that involve this type of antigen binding to red blood cells, and this is not normal. Okay, these are types of autoimmune diseases. So there are these classes of autoimmune diseases. They are called hemolytic anemia. So this means that you produce antibodies that attack your red blood cells and break them up. And once you break up your red blood cells, you become anemic. Okay, so you're going to have lower red blood cell count because you produce antibodies that broke your red blood cells. So this is called autoimmune hemolytic anemia. And this stuff gets more complicated because you don't have only one type of these types of anemia. Okay, so what happens here? In this case, your body is going to produce antibodies. Those are going to identify, they can bind to your own red blood cells. Okay, and the way that they bind, they use this fragment. Which one is this part of the antibody? Is it the point that binds, or is it the constant region? That's the binding part. Okay, so when you look at your antibody here, the FAB region is the top part here that binds to the antigen. Okay, so here you have this FAB region that recognizes something on your red blood cell, and it can bind to the red blood cell. Okay, and the interesting thing about this is that if you are producing antibodies of the class IgM, remember that's the pentamer, right? If you're producing antibodies of that class, you're only going to have this type of binding when the overall temperature is low. Okay? So if you live here in Florida, you wouldn't have much of a problem of your autoantibody binding to your own red cells, because most of the time the weather here is quite warm. But if you live in north, climate, if you live back in New York or something, the temperatures go sub-zero quite often, right? Then you need some some medical treatment, okay? Because when your body gets exposed to cold temperatures, the cold temperature facilitates or increases the binding affinity of your IgM against your red blood cell. And if IgM binds your red blood cell, you can form agglutination, right? So agglutination, that is going to make blobs of stuff in your blood, and that's going to clog your little capillaries, okay? So this type of disease is called cold agglutinin, okay? So this is called here, cold temperature agglutination. They have a specific name for that. Okay, here we go. Cold agglutinin disease. This is what it's called. So this here involves your IgM, okay? And since you have a cold agglutinin disease, you have another even more like rare type of disease that is called warm hemolytic anemia or warm agglutinin disease. That one is mediated by IgG antibodies. That's the monomer guy, okay? So basically these are diseases that are induced by binding of autoimmune antibodies to your own red blood cells. Okay, so you have cold-induced, that's IgM. You have warm-induced, that's IgG antibodies. Okay, yeah.
So, because I know you said that the kidneys, they regulate like the red blood cell count, so it would just hurt the kidneys.
You're going to have a lot of problems with the kidneys with these types of diseases. Because when you have agglutination, right, you're going to break, you also have a hemolytic part that you're breaking up the red blood cell. You're going to have the iron part of the red blood cell, the hemoglobin that is going to go in the circulation, and that is toxic to the kidneys. And you're going to have these blobs. They're going to be getting filtered, and they can clog up the glomerulus as well. So yeah, you can have a lot of kidney problem with this type of diseases. Yeah. But yeah, this is quite interesting. Like, autoimmune, cold dependent, weird stuff. Okay. All right. So here, so that was an example of disease, right? Diseases. So here, more normal function of antibodies. Okay. So remember we mentioned before the ADCC, right? This is your antibody-dependent cellular toxicity, right? Actually, cellular cytotoxicity is like a double thing. Okay. So what's going to happen here? You need two things, okay? So first thing that you need to have is the cell that is going to do the killing has to have a receptor that is going to recognize the constant region of the antibody. So this guy here is your Fc receptor. This is your Fc receptor. Second thing that you're going to have is that your killer, let's say this guy is a natural killer, it has to have toxic stuff in little vesicles, like ready to be dumped at the surface of whatever it is trying to kill. Okay? So this thing has to have the receptor, and it has to have toxic stuff ready to kill whatever it interacts with. Okay, so that's antibody-dependent cytotoxicity. Okay. So another function of antibodies, as I mentioned, is degranulation. So we're going to see this function basically only like related to IgE. Okay, so what happens here is that you have mast cells that have a specific receptor on the surface that is a constant region receptor, but only for your E, so is epsilon, is a receptor for epsilon, constant region of antibody. So this thing is only going to recognize the constant region of your antibody that is IgE. And IgE is going to be produced when you have worm infection, helminth infection, or allergies. Okay? So when your mast cells recognize that concentration, they are going to dump out their granules here, and cause damage to the tissue and also allergic reactions. All right. So got it, mast cell degranulation? All right. And this we have seen before, contribution of antibodies to the complement system. So membrane attack complex formation, we have seen that before. What is the name of the first protein that binds to the antibodies on the complement? The cucumber flying around with few legs, like 6 legs. Complement, you can guess, it starts with C. C, that binds to the antibody constant region. IG or IgM, IgG or IgM. Okay, I'll give you another clue. It looks like this, and it has little legs. Yes, I heard it. Yes, C1, go for it, what C1. Remember, it's one QRS, you got the RS in the middle here, right? Yeah, so C1 for your classical pathway, right? All right. And what is the C3 convertase of classical pathway? Oh, no, C4B, C2A. You see how confusing that is? C4B, C2A. Because this guy is going to cut C4, is going to cut C2, then this thing is going to opsonize and get there, right? This guy is C3 convertase, then that's C3 over here. So if you put C3B over here, then is when you get your C5 convertase. After you write it down a few times, then it's going to be like second nature. All right. So here, functions of all these different classes of antibodies. Right. So this is a lot of information on this slide, and it's quite important for you to know. Okay. IgM, that is the first one that is produced. It is on the surface of your B cells that are mature but still naive. Okay. Once you have exposed the B cell to antigen, you're going to start producing IgM. So that is the first antibody that is going to be produced in active and recent infections. Okay? IgG, and remember this guy is pentamer. IgG, that's the most abundant in the circulation. It's very important for all those effector functions of the antibodies. Okay, so you have here your activation of the complement, you have opsonization, ADCC, neutralization, and interestingly here, your IgG is the only antibody that crosses the placenta. So it is small enough and it has the proper structure to cross the placenta. And it's not just a matter of being small enough, because you have the other antibodies that are also monomers, like IgA in the bloodstream is monomer, but IgA does not cross the placenta. Okay? So IgG does. So that means when the baby is born, and usually crosses at the end of the 3rd trimester. So if you have a full-term baby, by the time the baby is born, it has IgG antibodies from the mother. So it has what kind of immune defense is that? It is natural. It's passive and it's natural. Yeah, right. So it got naturally from the mother, and it's passive because the baby did not produce those antibodies. Right? Passive, natural. So a baby is going to have those antibodies for some time, not forever, right? Because then the baby will have to develop their own antibodies. Right? So IgA is important for mucosal protection. IgA and IgM, but IgA more than IgM, okay? Okay, IgD, the only thing that we know is that it directs the naive mature B cells to secondary organs. That's all the function that I know of IgD, okay? And is never secreted. IgD will not be secreted, okay? And then IgE is against those pathogens and is also highly inflammatory. Okay, so here is a table from the Janeway, from the new book. Okay, what I like about this is that it shows you the types of antibodies that are important for classical pathway of complement system activation. So here you see your IgM and your IgG, even not all types of IgGs, but a lot of IgGs. Okay, and here you see. Okay, forget about this guy. Here, you see placental transfer. Look at this. Only IgGs get transferred to the placenta. Okay? And let me see, binding to Fc receptors of phagocytes. Look at that. This is the function of opsonization. Okay, opsonization. Look at the antibodies that can cause opsonization. Okay? Okay. Let me see. This one here, look. The ones that are going to activate the granulation, okay, mast cells, basophils, only this guy over here. Only IgE for the granulation. Okay. Forget about this too. Okay. Oh, this is a cool antibodies poem that you can read at your leisure. Okay. All right. So finally, we get here to the lymphocyte development. Before we get into this, do we have any questions? Yeah.
Isn't there like a condition in like pregnant women who have like O negative blood and like how that?
Yeah, that is hemolytic disease of the fetus. I do think I have sessions that I covered that. But yeah, I'm not going to cover that right now. Yeah, but we'll get to that. Okay. All right, so let's see here on lymphocyte development. Okay. Yeah, we'll get there when you get to activation of lymphocytes. Yeah. All right, so lymphocyte development. What is happening here? We're going to know how those lymphocytes, especially B cells and T cells, how is it that they're going to get their very unique receptors? Okay. Basically, they go through the same process of gene rearrangement. So this is when it's going to start talking about, rearranging the DNA for every one of those cells that are going to come out of the bone marrow and the thymus. The DNA is going to be rearranged. It's not going to be the same DNA as you have inherited from your parents. It's going to be different. The DNA of those cells is going to be different. Okay? Each clone is different. Each clone is going to be unique. Okay? So you're going to have two types of chains, two types of gene rearrangements. They're going to make the receptors of those lymphocytes. So you're going to have heavy chains, okay, and you're going to have light chains. So both receptors, okay, both T cell receptor and B cell receptor, they're going to have heavy chains and they're going to have light chains. Okay, so that process of rearrangement is the same. Okay. Now we're going to understand why that process of rearrangement and recombination is important. Okay, and then we're going to understand how that goes. So what's the idea here? The idea here is that once you pass this level of innate defense, so first is a barrier and then the chemicals, right, mucous membranes, and then you have innate defense that is related to your natural killer cells, right, and the other cells that are produced either by the liver, or you have those myeloid cells of innate function, right? And then you have also basic antiviral responses like interferon production. These are all part of your innate system, right? Now, your innate cells are going to communicate with your adaptive cells, and they are going to teach your adaptive cells to produce specific antibodies, differentiate, and to start producing specific types of defense. Okay? The way that those cells communicate is via production of cytokines. So chemokines and cytokines, they're basically falling in the same bucket of molecules that are produced from certain cells to communicate what other cells should do. Okay? All right, so let's see what is going to happen here. So here, at first you had your innate immune system that tried to destroy your pathogen, right? So here, first thing that's going to happen, you have here your phagocyte that recognizes the pathogen using a PRR, right? PRR. And this thing was a PAMP, right? And then you also have PRRs inside of vesicles, and that thing all, processed the pathogen. Now your phagocyte chopped up the pathogen and placed it on a surface or on a molecule. Okay, in this case here, this is MHC class 2 molecule. Okay, because this pathogen came from outside, got processed, put on the MHC class 2, and then this thing is going to go and show up on the surface of this cell. So this guy here can be, what kind of cell is that? What is the name for the cell? This phagocyte over here. Let's say it's a dendritic cell. Innate cell from from myeloid lineage, right? So this guy here is going to present a pathogen on MHC class 2, and this thing is going to interact with a T cell. And this is a naive T cell because it's going to recognize stuff on MHC class 2. Okay? So now you're going to have interaction of these two cells, and you're going to have activation of your naive T cell. Okay? So what's going to happen? This antigen-presenting cell, when it presents the antigen to your, not antibody, okay? Antibody is only B cells. When it presents the antigen to your T cell, it is also going to produce certain cytokines that are going to teach your T cell to react a certain way based on what it ate. Okay? So let's say here that it ate a virus. Okay, so if it ate a virus, it's going to produce certain inflammatory mediators or cytokines that is going to teach your T cell to differentiate into a Th1 type of cell, right? Because that's the type of cell that is going to fight off internal pathogens or viruses. Okay, so this interaction here is going to teach that cell how to behave. But in order for this thing to start happening, you have to have a receptor on your T cell that is capable of recognizing specifically that antigen right there. Okay, so let's see how that happens. So now we're going to have the communication between innate and adaptive systems. Okay, let's see how that happens here. So let's see here, overall picture. In the bone marrow, remember all of your myeloid cells develop in the bone marrow, and they come out of the bone marrow ready to work, right? But also you're going to have in the bone marrow the development of your lymphoid lineage, right? So in the bone marrow, you had those specific types of signals that trigger a cell to develop into a lymphocyte, okay? This lymphocyte can either be a B cell or a T cell, right? So what's going to happen? The B cell is going to do all of its development in the bone marrow, then it's going to come out of the bone marrow as a mature but naive T cell, and is going to further, a B cell is going to further differentiate in some secondary lymphoid tissue. Your T cell is going to come out of that bone marrow not as a T cell, but it's going to come out as a precursor of a T cell. So this guy is going to go to the thymus, and in the thymus, it is going to differentiate and undergo gene rearrangement and become a T cell, and then come out of the thymus as mature but naive T cell. Okay? For the B cell, all of the rearrangement, the gene rearrangement and the formation of the receptors, everything is happening in the bone marrow. Okay? And then for the T cell, everything is going to happen in the thymus. Okay? And this is why you have your thymus also as a primary lymphoid organ. Okay? So after it comes out, it is mature but naive, and then it goes to secondary lymphoid organs and sits there and waits for the proper pathogen to show up. Okay? All right. So after it sees the pathogen, it becomes experienced. Okay, it's going to become effector T cell. Okay, what kind of T cell? Well, we have several types of T cells that come out of there and be effectors. You have the ones that are helpers. Those are some effectors T cells. You have the ones that are killers. Those are cytotoxic T cells. You have the ones that are memory cells. Those sit around and wait to be activated. Right? That's what's going to give you a very good secondary response to seeing the same antigen. Okay? Besides all that, you also have those suppressor cells. Remember those guys? T regulatory cells. So those are all effector cells. Okay, so these guys here are cytotoxic. These guys here are helpers, and these guys here are regulatory cells. And then you have your T memory cells. These are all effector cells. Okay? All right, so now let's look here at the side of the B cell. After they see the antigen, they're also going to become experienced effectors. What kind of B cells are you going to have? You're still going to have some memory B cells. They're going to come out of there, and they're going to sit around and wait to see the same antigen again, right? You have some memory B cells. And then you're going to have plasma cells that are going to produce different classes of antibodies depending on what the information was on the initial infection. Okay? So here, let's say you have a viral infection, you're going to produce IgG. If you have a bacterial infection or any type of infection, you're going to start producing IgM. And then maybe later on it's going to class switch. Okay, and so on and so forth. Okay, so those are also all effector plasma cells. Okay? All right, so here is just a bit of a review that we had this slide before, right? So lymphocytes, types of T cells. Okay, remember CD4 positive T cells are going to recognize things that are presented on MHC class 2. One important thing about the T cells is that they are going to see a little piece of antigen that is stretched out on this MHC class 2. Okay, so this is your MHC class 2 presenting with little hands. And this little piece of protein is a little twig, is stretched out. It is not a 3D shape like this. It is not something that is whole. Okay, it's only a little piece of stretched out protein like this. So the T cells, it doesn't matter if it is T helper or T cytotoxic, is going to recognize something that is presented in a 2D form. So it recognizes 2D. It doesn't recognize 3D whole thing. Okay? All right. So that's how your T cell is going to recognize stuff. And this again, we had this slide before, so just to remind you to revise these areas here. Okay. Now, T cell development. This is when the cool stuff starts, right? So this thing is about the lymphocytes. So it's called lymphopoiesis. I took these slides from osmosis, okay? From the platform osmosis, all right. So what is going to happen here? There are three important things that this process has to accomplish, okay? First thing is that it's going to produce a unique receptor. So each one of those cells is only going to be capable of recognizing one epitope. Okay, only one epitope. Second thing that this process has to accomplish is that you don't want to recognize epitopes that are present in your own body. Otherwise, you are going to have autoimmunity. You are going to produce cells that can destroy you, and that's not a good idea. Okay? You only want to produce cells that are not going to recognize stuff that is in your body. Okay? So you have to get rid of any progenitor cell that recognizes self stuff. Very, very important. If you don't get rid of them, you're going to have autoimmune disease. Okay? Now, if the cell is capable of producing the unique receptor and it also does not recognize self stuff, okay? What is going to happen here is that you're going to have the green light to develop a whole clone of that one particular cell. Okay? So now you can come out of the primary lymphoid organs and you're free to go to the periphery and start patrolling, trying to find the antigen that you were created to recognize. Okay? All right, same story with B cells, same story with the T cells. Okay. All right, so what's happening here? To become a B cell, right? Everything is going to happen here inside of the bone marrow. They start from the same hematopoietic stem cell. Then you have a common lymphoid progenitor cell. Then you're exposed to those cytokines and factors that tell it to go towards the B cell lineage. After that, you mature, you come out of the bone marrow, and you go to secondary organs. It could also go to mucosal-associated secondary organs. It's not shown here. Okay? And after you find your antigen, you become effector cell that is called your plasma cell. Okay? And then plasma cells are going to shoot out antibodies. All right. That's the story with the B cell. Now with the T cell. The T cell goes as a precursor to the thymus. In the thymus, is going to become a thymocyte. After it undergoes all this development of receptors, it's going to become a mature, naive T cell. Then it's going to go to the circulation. Okay? All right. So in the thymus, it's going to become a thymocyte. Okay. Now, let's see what happens here with the thymus. When you are born, you have a huge thymus. It's really, really big. I think I had some picture of this baby, like they have the picture of the like transparent baby with a huge thymus, like in proportion is among us. Okay? As you get older, the thymus starts to shrink. Not only it shrinks, but it transforms itself into fat. So the parts of the thymus that are important for T cell development, those parts kind of disappear after a certain age. And then you are left with this tiny thymus that is just fat. So what's going to happen? Or what's not going to happen?
Development of T cells.
Exactly. So now, if you only have a thymus that has fat in there, fat is not going to do a T cell gene rearrangement. So you have a big problem because you're not going to make new T cells. So after a certain age, you have to rely on your memory T cells in order to fight infections. And some of your memory T cells are not going to live forever. So what happened to the elderly? They get sick. They're more prone to develop infections, right? Remember during the COVID time when the vaccines just came out, and like, okay, let's vaccinate the elderly first because we got to make sure that they just don't die if they're exposed to the virus, right? Because their immune systems are already low, right? So yeah, it's a big problem of losing your adaptive immune system after a certain age. Okay, so you call that progress the involution of the thymus. Okay, so cell-mediated immunity, meaning your adaptive immunity, is going to go down as you get older and older. Okay, all right. So here, let's see what is going to happen here. Remember that for T cells to see antigen, they have to see it presented on MHC, right? Now, we're going to look at how the genes are rearranged, because both of those rearrangements are similar, right, for B cells and for the T cells, okay? And both B cells and the T cells, they undergo this negative selection process. So that means if a cell recognizes self-antigen, the cell will be killed. That is your negative selection, okay? Both B cells and T cells go through that. There are some things that are different between the developments of B and the T cells. So things that are different. First thing is that the T cells have to migrate to the thymus, otherwise they're not going to develop. Okay? T cells have to recognize things in 2D presented by MHC, and that tells you something else, like how is it that the B cell is going to recognize antigen? Think about it. Okay, here's the antigen. If you have a cell that goes in there and tries to recognize the antigen, how is it going to do it? It's going to look at it and say, I think it is a bacterium. No, it doesn't have eyes. It's going to go like this. Ah, you recognize it. What does that mean? Yeah, it's a receptor. There's a B cell receptor. The B cell receptor recognizes in 3D the whole thing. Okay? It does not depend on MHC presentation. It recognizes the whole thing. Okay, the whole molecule, the whole bacterium. Okay, the B cell recognizes 3D. The T cell only is going to recognize the stretch of protein presented on the MHC. Okay, different ways of recognizing. So that's a very important distinction between recognition of the B cells and the T cells. Okay, so because the T cells need to recognize the thing in 2D and presented by MHC, the T cells will have to undergo a further checkpoint to make sure that it recognizes the proper MHC that is going to present the antigen. Okay, so the T cell is going to have one extra step for proper recognition of MHC. Okay, so not only it has to undergo the negative selection, it will have to undergo a positive selection for MHC recognition. Okay? All right. So here is the important difference between B and T cells. All right. Another important difference is that your T cells are not going to secrete their receptors. The receptors of the T cells are always going to be on the surface of the T cell. Okay? So the receptor, the T cell, is not called antibody, it's just called T cell receptor. Okay? B cells are the only ones that are going to secrete those antibodies, right? Another thing about the T cell receptor is that it's not going to change after it matured in the thymus. So the way they come out, that's how it is. With the B cells, there is changing of the receptor. It can become any of the other classes of antibodies, right? It comes out as M or D, but then it can class switch and become some other kind of antibodies that's going to shoot out. Okay, so that is not going to happen with a T cell. T cell has a receptor, that's it. Okay? All right. So let's see here, how is it that the T cells are going to develop? So you have here your precursor cell, precursor lymphocyte that came from the bone marrow and is going to go to the thymus. So this whole thing here is the thymus. Okay? So it's going to get inside of the thymus, like more or less here in the middle, okay, between the cortex and the medulla. So here cortex and the more inside, this is showing like basically everything is closer to the cortex. But let's say this is more to the inside, okay. This is the between the cortex and the medulla, this is how your lymphocyte precursor comes in there, okay. Then what is going to happen to it is going to go under the capsule of the cortex. By the way, the thymus has one of the strongest capsules of any tissue in your body. A very, very strong capsule. Because you don't want crazy T cells to come out of there and start recognizing your own tissues. That's going to be deadly. So they have to keep these cells encapsulated until the final stage of development when they are safe to come out. Okay, so it's a gel of T cells over there. T cell school. Okay, all right. So that's going to happen. The T cell gets in here. It goes to this space under the capsule, and it starts to develop. Then it goes through the cortex, and then it goes to the medulla, and then finally it will come out. Where is it coming out from? Oh, it's not even showing it coming out, but it will finally come out. Okay. Okay. So here there are a couple of points here that have also a clinical correlation, right? In patients that have this syndrome that is called DiGeorge syndrome, you have underdevelopment of the thymus. So when the baby is born, it doesn't really have that big thymus. It has a thymus that looks more like an old person's thymus. Okay, so underdevelopment. So what's going to happen is that the baby is not going to be able to produce those T cells at the rate that it's supposed to. So either they have very little amount of T cells or they have no T cells at all. That's even worse. Okay, so these patients, they're not going to have adaptive immunity. Okay, so that is a form of disease that is actually very deadly. Okay, because the patient is very much susceptible to all forms of infections and diseases, right? All forms. So that's your DiGeorge syndrome. If you're born with underdeveloped or non-developed thymus. Okay. So how is it that the T cells live their lives? Well, remember, they go to the thymus and they mature, then they come out of there being mature, and they go to secondary lymphoid organs, and those are the lymph nodes. You have the spleen that filters the blood, you have the gout, right, the gut-associated lymphoid tissue, and they don't stay there. They like to travel. They get in there, they don't find anything, they go back into the circulation, they travel through the lymph, they travel through the blood, and they go all over the body, and they keep on traveling. Okay, and this is also another difference between B cells and T cells, because B cells tend to just go to one place and stay there for a longer period of time. And the T cells like to travel a lot. Okay, B cells also travel, but not as often. Okay, these guys keep on traveling. All right. Then what's going to happen here? Okay. Remember that when your T cell, okay, let me see here. Okay, when your T cell reaches here, your thymus, okay, remember you have these two areas. You have the cortex, subcortex space, and you have the medulla, okay. When the cell goes there, it starts to get information from the tissue that is already there, so you call that epithelial tissue, epithelial cells that are present here in the medulla, okay? And inside of the cortex, you're also going to have antigen-presenting cells, specifically dendritic cells that are sitting in there, and they have the job to present things to your developing T cells, okay? So your dendritic cells, remember, those are the professional antigen-presenting cells. They have the job of teaching the T cell what it should see and what it should react to, okay? So is going to be present there, and is going to be showing all sorts of antigens to the developing T cell. Okay? Okay, remember here, your T cell sees in 2D, okay? So your T cell receptor that has two parts to the receptor is going to recognize MHC presenting molecules, and then they're going to recognize the antigen, okay? So this is always happening in the timers. So how is it that your T cell is going to develop the receptor? Okay, this is the part that gets interesting. Because here, it tells you that you inherit a whole bunch of genes from mom and dad, right? Now, when you're looking at your T cell receptor genes, you have one bunch of, a lot of genes that are going to make your beta chain. The beta chain is the bigger chain, okay? The heavy chain, comparison to the heavy chain of the B cell antibody. So you have a beta chain, and then you have another gene that is for your alpha chain. So let's say, how many genes do you get from your dad that are going to code for beta chain? Okay, everybody took genetics? Yeah, yeah. You get half of your chromosomes from mom and half of the chromosomes from dad, right? So you're going to get one chromosome from dad that encodes for beta chain. You're going to get one chromosome for mom that also encodes for beta chain. Okay? Then you're going to get the chromosomes also from mom and dad that encode for the alpha chain. Okay? So you're going to have two. You're going to have, let me put here a color coded. You're going to have here a chromosome from mom that has beta chain stuff in it. You're going to have a chromosome from dad that also has beta chain genes. Not only one gene, that's genes, okay? And then when you look at the alpha chain, you also see the same thing. You're going to get this chromosome and that chromosome, and they're going to have genes that encode for the alpha chain, okay? So then what's going to happen? When you look at the beta chain itself, the beta chain is made up of 3 portions. Okay, so to make your beta chain, here's your beta chain, like this, you're gonna have to have 3 pieces. One piece is called V, the other piece is called D, and the other piece is called J. Okay? So in order to make a beta chain, you're gonna have to choose the pieces. Each one of those genes that are coming from either mom or dad, they are going to have many options for each one of those segments. Okay? So here, the picture is showing you that, let's say, from mom, you're going to have all different options for your V, all different options for your D, and all different options for your J. So basically, it's like you have your closet of clothes that are coming from mom's side of the family, and you have to choose an outfit that is going to be your beta chain. So you can go in there and you can say, okay, I'm going to choose this shirt with those pants, with this shoe, and now you're going to have your beta chain that is going to look like this combination of outfit. Okay? Now let's say that instead of that, if you want to choose something else that is, say, you're going to choose this gene with those genes and these over here, now you're going to have a different beta chain that is unique with those combinations of gene segments. Okay? So this is a way of recombining the portions to make your entire beta chain. Okay? So this is what you call recombination. Just based on what you see over here, how many possible combinations can you get out of these outfits? This is testing your math brain. You remember independent terms? Yes? How many people have no idea what I'm talking about? Okay, let's see. Independent terms. How many independent terms here? 4. How many independent terms here? 4. And here? 4. What do you do with independent terms? You multiply them, right? Does it sound like Greek? Hopefully not. So 4 times 4 is 16 times 4 is, I trust you, 64. 64 possible outfits you can get out of this closet. Okay? So based on these small segments of genes that are going to make your beta chain, you can end up with 64 different clones only for the beta side of your T cell receptor. But you're only dealing here with your chromosome from your dad. You also have all of those from your mom. That's a lot of combinations, right? Because you're also going to have 64 that are possible from your mom. So you're going to have a total of 128 possible T beta chains, okay? And on top of everything, you don't only have 4 segments of this or that. You have like 60, 40, 100. It's a lot of combinations, okay? So now let's see what happens here. First thing that's going to happen on selecting the pieces of genes is that you're going to get here for the beta chain always arranges first, okay? So beta chain, you're going to start with, where is it? Okay, beta chain starts first. Let's go to the next one. I lost my slides. Okay, 1 second. Okay, let's get you some points before you leave, right? I'll go back to those slides. Okay. Okay. Most distinctive property of a mature T cell is what? Where the T cell receptor recognizes the peptide antigen only when combined with MHC molecule, right? MHC molecule that is presenting it, that is also part of the person. What is your answer here? I have 2 people there telling me something that is not correct. You need more time? No? What's your answer? A. It has to recognize the stuff that is on MHC, so MHC restriction. Okay, I'm going to stop here, and for next time I'll go back to the slides that I just jumped.